I hope you guys can see my
Yes,
slide.
yes, we are able to see
Great.
your slides. Please go ahead.
Okay, great. Hello everyone. Today I'm going to talk about the rice disease bacterial panicle blight, which is a major the project in my lab. Actually, we are working on both the pathogen in terms of the virulence factors and the rice disease resistance to the bacterial panicle blight. But today, my presentation will be focusing on our research on the disease resistance to the bacterial panicle blight. Uh, okay. So first, what is the bacterial panicle blight? This rice disease is caused by the plant pathogenic bacteria, the Burkholderia glume and the Burkholderia gladii. Uh, Between two bacteria, the Burkholderia glume is the prevalent and actually is considered as the major the pathogen. And this disease is severe in the unusually hot weather and high night temperatures. And this disease can cause the yield losses from the 15 to 80%. And the severe epidemics and the severe yield losses from this disease occurred in the mid 1990s and the early 2000, 2010 and 2011 in the southern United States. But nowadays, this disease is also to become a serious problem in the other parts of the world probably due to the climate change. And uh, unfortunately, most of the commercially the valuable varieties in the United States and also the other part of the world, that they are susceptible to this disease. And but however, still we don't know much about the disease cycle of this disease, how this disease they initiate and how they disseminate and they multiply. Those kind of cycles not been known very well. So, but we only know what we the guess is that probably this disease is a seed-borne disease. So the pathogen is present in the seed. And so that planting the clean seeds would be maybe the improve the disease management, but still we are not clear about that. And then regarding the disease cycle, we can think about the three major stages, including the establishment in the host plant and the deployment of the virulence factors, uh, the for the symptom development and the dissemination of the pathogen, but none of these stages are not elucidated clearly so far. And then for the disease management, because this disease is caused by the bacterial the pathogens, so the fungicide is not useful, and so the no chemical product available for the management of this disease. The antibody compound called the oxalic acid is used for as a chemical agent in the other rice growing countries, but this chemical is not allowed to use in the United States. So we need to find out the alternative disease management strategies in addition to rather than the chemical agent. The probable chemical agent that can work would be the copper-based chemicals, but according to our tests, the copper-based chemicals caused the phytotoxicity in the rice. The rice tends to be very sensitive to the copper, so this option may not be recommendable, I think. So we should find out the alternative approach to the disease management. One of them is uh, to develop the more resistant rice varieties. So, however, most of the conventional varieties in the southern United States are susceptible to the bacterial panicle blight, except that several the varieties or lines to show some the moderate resistance, including the medium grain variety Jupiter and then some mutant line, including the LM1, show the moderate resistance. As you see here, this is the plot, growing the different lines, and then the Jupiter and then the LM1 shows kind of the more resistance than the susceptible variety, Trinas and the Vanguard. So our idea is to just characterize the genetic loci responsible for this moderate resistance in the Jupiter, and try to utilize those they identified the loci to improve the disease resistance of the U.S. The susceptible U.S. varieties. And so the first we did the genetic study for the QTL mapping using the mapping population, the recombinant inbred lines derived from the moderate resistant variety Jupiter, and then the highly susceptible Trinas. And then we tested, generated about the 300 recombinant inbred lines from this cross. And then this picture shows the phenotypes of the disease, highly susceptible Trinas, the moderate resistant Jupiter, from the inoculation of the artificial inoculation of the pathogen. We could see some distinct difference in the disease, but sometimes the difference was very obvious. But some environmental condition, the Jupiter also shows quite the high level of disease. So it depends on the environment. And just grow the mapping population in the field, and then just the phenotype in terms of the disease resistance, and also the agronomic traits, and also other disease resistance. And we found that the phenotype related to the bacterial panicle blight is quite closely related to the days to 50% heading phenotype, which basically means that the flowering time, it was very closely related. And also closely related to the another rice disease, major disease, sheath blight, caused by the fungal the pathogen. So all the three traits, the bacterial panicle blight, the sheath blight, and days to heading, they are very closely related according to the multi-year the field trials. So the sheath blight, bacterial panicle blight, and then the days to heading was very closely related. And then we did the QTL mapping using the three approaches in the genotype. The first, the conventional random single sequence repeat SSR markers, and also we genotyped using the CASM markers for the SNPs, and we also employed the QTL signal analysis for find the mapping. And then from this the genetic mapping, we found that all the three traits were majorly controlled by the single locus on the top of the chromosome 3. In case of the sheath blight, the 3.1 location of this sheath blight 3.1, and then the bacterial panicle blight 3.1 here, the 2013 data, and then the days to heading phenotype, the QTL, the 3.1, all of them just located in the same locus on the chromosome 3. In fact, the sheath blight 3.1 and then bacterial panicle blight 3.1 was previously reported by another research group in the 2010 from the different rice variety called Taking. So this is the map just showing that the location that on the top of the chromosome 3, all the one locus just plays a major role for all three traits, including the bacterial panicle blight and the sheath blight and the days to heading. So they are just concentrated into the one locus. And the 2012, the data, we mapped the another the locus very close to the 3.1, but this one also very closely located into to the 3.1. And the next approach is that we just tried the QTL signal analysis, using the eight the resistant, most resistant recombinant inbred lines, and then 7 most susceptible recombinant inbred lines, and compare the whole genomes of them. And then from this analysis, we could identify the 8 QTLs with an average interval of the 2 megabase. So which means we could actually see that the candidate genes in each locus. And then out of 8 QTLs identified, uh, the most significant one was also identified from the top of the chromosome 3, indicating that the major QTL from the conventional the mapping also identified from the QTL-seq analysis using the selected recombinant inbred lines. And so those the 8 QTL from those 8 QTLs, there are about the 2,500 genes located in those QTLs, but surprisingly, only 9 were predicted to be involved in the plant defense, signaling, or the flowering. Especially in that the major QTL area, we found 3 the most the reasonable genes. The one of them is the mad box gene, which is known to be a regulator for the days to heading phenotype, which is consistent with our the phenotype. And the 2 others are the defensin gene and then the chitin-Ellis, the binding protein. They might be the responsible for the resistance phenotype of their locus. And then the one question was that because the disease resistance for both the bacterial panicle blight and sheath blight and the flowering time is co-located, so we consider the possibility that increased disease may be probably due to the delayed flowering, maybe avoidance of the pathogen infection. However, we identify at least 2 the lines that decouples the phenotype of the flowering time and then the disease resistance, which means the most of the resistant lines shows the late heading phenotype, and most of the susceptible lines shows the early heading time, but these 2 lines is not following that pattern. So indicating that at least probably the disease resistance is not the 100% due to the early the flowering time. So, however, we just decided to do the another set of the genetic study using the different mapping population, the recombinant inbred line population derived from the same resistant source, but the susceptible source was the Bengal at the time. The Bengal is the same, the medium grain variety, that like the Jupiter, the temperate Japonica type. So Jupiter and the Bengal, they are different in the disease resistance to the bacterial panicle blight. However, they have the similar the heading date, the phenotype. So using this mapping population, we think that the effect of the heading date would be minimized. So from this mapping population, we get a phenotype and then the genotyping using the genotyping by sequencing approach. And then from this mapping population, we identified the 7 QTLs for the BPB. But however, all of the QTLs identified did not contribute very much to the bacterial panicle blight resistance. So we could not find the very strong the locus like the 3.1 locus. However, the one of them, 3.3, is very close to the 3.1 and 3.2, but there's a similar location. So then which means there's some the limitation in the using the current resistant rice cultivar to increase the disease resistance by the conventional breeding. So we researched the literature, and then there's only one on the major QTL for the bacterial panicle blight from the different type of the rice, the indica rice called the Calais. So that major locus found from the Calais is located in the chromosome 1. So that was identified in the around 2018 by the Misobuchi's group, and they what they did was that they just crossed the discale into the susceptible Japanese cultivar, the Hitomibore susceptible one, and did a series of the backcrossing to generate the new isogenic line for the introducing of this major locus into the Hitomibore, and they could just improve the resistance from this susceptible phenotype into the quite a strong resistance through the introduction of the RBG2 locus into the susceptible variety. So our approach currently we are doing to improve the US rice is that's one of the approach that introducing this RBG2 and the 3.1 locus from the US variety all together into the susceptible rice to improve the disease resistance. That is the primary, called the pyramiding approach that to just the kind of the additive the effect for the additive effect of the resistance by accumulating the resistant the trait into the susceptible rice through the conventional breeding process. But that takes a lot of time, and we are not sure how effective it would be. So at the same time, we also have tried the testing the many other the materials that can be used as alternative the agent for the disease management. So we tested a variety of the natural materials and then the biological materials, including the ascorbic acid and then the acetylcysteine, chitosan, and then some biological the materials identified from our lab, and tested them in the field, in the greenhouse, and so this is kind of the scale that we are using to evaluate the disease, and so from the 0 to the 9 scale. And this is a kind of representative data that we obtained from the testing in the field. And among those, the materials that we have tested, many of them show the significant disease suppression effect. But out of them, the biological agent, 8257, shows the best the activity in the cultivar, susceptible cultivar Bengal, and also the another the susceptible cultivar, the CL111. So in both cultivar, this agent shows the most promising activity. And this is the plot just showing that the effect of the 8257 treatment compared to the non-inoculated clean plot, and then the buffer treated kind of control plot. So it shows that, as you see, that all the panicles is quite clean as the non-inoculated the plot. And the next question is that the foliar application of this agent maybe takes a lot of time and a lot of the labor. It's not really cost efficient. So we tested if that agent will be the effective through the sheet treatment actually. So what we tried was to just to treat the rice seed before planting. We just treat the rice seeds into the bacterial suspension of the A257, and then they treat them on the field. And after they grow, we inoculate the pathogen for the bacterial panicle blight, and also we tested the another the rice pathogen, fungal pathogen, the Rhizospora solani, which caused the sheath blight, the another major rice disease. And then for your information, most of the slides showing the effect of the 857 were prepared by the PhD student, the Jobel Bruno. So I would like to credit for this part to her. And this is the data showing that the suppression of the 857 compared to the non-treated plants. And in the 2020 and the 2021 and the 2023, we got the similar pattern. Even though in the 2021 the disease pressure was very low, so but still we could get the kind of significant reduction in disease. And we also tested the sheath blight, another major rice disease, and then in the greenhouse and the field. And as you see here, we could see the very significant reduction from the rice grown from the seed treated with the 8257. And in 2020, in the sheath area, we could not see any significant reduction. But if you just see that the panicle, evaluate the panicle, we could see the much lower symptomatic panicles in the seed treated the plant. And also the similar pattern also observed in the 2023. So throughout the 3 years of the field trials, we could see the effect of the 8257 in both bacterial panicle blight and the sheath blight. And also, even though I don't show that result in here, but we also observed the reduced disease in the natural, the Cercospora, the narrow lip, the spot disease caused by the Cercospora, the gentiana. So then currently we are working on the mechanism of that sheet to treat the effect of the 857 sheet priming. And so to see that initially we did the RNA sequential analysis, comparing that the seed without treatment. Without treatment means that just buffer treated and then the bacteria treated, and then grow the plant from those seeds and then inoculate them, and then compare the gene expression profile between two, comparing between the treated, untreated plant and also inoculated and the uninoculated the plant. So we just compare the four different comparisons, but the most meaningful the comparison was the inoculated plants between the seed treated or untreated plants. So because both of them show the disease, but the treated plants show the less disease, and why those plants show the less disease. And then according to our initial RNA sequence data analysis, we found that the carbohydrate, the metabolite, metabolic process, the genes involved in this process, so actually induced more in the treated plant, and probably it's just consistent with the previously known the information that the plants under the pathogen attack undergo so changes in the carbohydrate metabolism to support the defense responses. For example, and especially the plants treated with the PGPR, the plant growth promoting bacteria, actually increase the carbohydrate metabolism, which caused the increase of the energy reserves and the synthesis of the defensive compound and the regulation of sugar signaling and the antioxidant systems. So, but that's the data from the first experiment, and then we are also doing the additional RNA sequencing and then the qRT-PCR experiment to get more information and the confirmation of this result. So the summary of this study, we are studying on the disease resistance of the rice to the bacterial panicle blight. And the first approach was the conventional genetic mapping for the identification of the QTLs involved in the disease resistance. And the first mapping study, we identified the major QTL on the chromosome 3, which is overlapped with the locus for the sheath blight resistance, and then the day to heading trait, flowering trait. And then from the second population, we also the one major QTL close to the first previously identified QTL, and we are now the working on the integration of the RBG2 locus from the indica variety along with the major QTL that we identified into the US varieties to increase their disease resistance. And at the same time, we also working on the biological control agent 8257 for their efficacy in the increasing the disease resistance and studying the molecular mechanism underlying that induced resistance or induced defense pathways caused by the 8257 treatment. And then finally, I'd like to thank especially the John Ontoy, who just performed most of the genetic study in the first part of this presentation. My former graduate student, and now recently he joined the Clemson University as the postdoc. And then the Joe Bruno is current PhD student in my lab, and also the other the lab members and my collaborators in the Rice Research Station of the LSU Ag Center. And finally, I'd like to thank the funding sources, the Louisiana Rice Research Board, and then USDA NIFA, and then 2 grants from the USDA NIFA. And then thank you very much for your attention. And then I'd like to take any question on this project. Thank you. Any questions?
So, any questions for Dr. Jong? I'm sharing his contact details. If you have any queries, please contact.
Thank you.
Thank you.